So, good afternoon, guys. I hope I'm audible and my screen is also visible to you guys. Okay, so I'm Rishabh, and today we'll be starting with the week 7 of the host pathogen interaction NPTL course by Professor Himanshu Kumar. And in this course, we'll be discussing about the complement system. So, in this lecture, we'll be discussing about the complement system, its types, and how the process takes place. Okay, so starting with the complement system, some of the history basically. So, in 1890s, Jules Bordet basically described as a system that is heat labile. Okay, so in terms of heat labile was that it is sensitive to heat. Okay, so that substance whose activity in normal plasma could complement the bactericidal activity of the immune sera. Okay, so that means there is something that complements the activity of the serum proteins. So this statement was given by Sir Jules Bordet. Okay, so the experiment he performed was he actually uh, isolated the sheep anti-serum. Okay, so from sheep he took the anti-serum and he incubated it with Vibrio cholerae. So this is a causative organism of cholera. Okay, so and it is a comma shaped bacteria. So he incubated this and he observed that there was lysis of Vibrio cholerae when the anti-serum was incubated with Vibrio cholerae outside the body. Now, when he heated this anti-serum with nearly 55 degrees Celsius, now this anti-serum lost its ability to actually lyse these bacteria. Okay, and when he added fresh anti-serum to this particular solution, then the lysis activity actually restored after adding the fresh serum. Okay, so he suggested that there is a heat sensitive component in the anti-serum that is actually important for the antibacterial lysis of the serum. Okay, now after these of the experiments, Paul Ehrlich, in his work from Berlin, he conducted similar experiments, that is, using the serum and the heating of the serum at particular temperature, so that it because it's heat sensitive, it will actually lyse the bacteria. Okay, so and then he coined the term complement. Okay, so why he coined the term complement? He said that there is some components in the serum that actually complements the activity or the function of antibodies. Okay, that's why the complement name came through. So now this system that lies the bacteria is termed as the complement system. Okay, so this was a brief history how the scientist actually reached to the complement system, and now we'll be moving on to its basic introduction. So it is basically a plasma protein cascade, or the proteins that are present in the plasma that functions in a cascade manner that helps the antibodies and phagocytes to clear the microbes, promotes inflammation, and can directly lyse the cells. Okay, so there are certain proteins that are present in the plasma that function in a cascade manner. Okay, so what is a cascade? It basically one molecule activates the other molecule activates the other. So likewise, this is a cascade. Okay, so they help the antibodies and the phagocytes to clear out the microbes, to promote inflammation, and can directly lyse the cell by forming the membrane attack complex. Okay, these are soluble proteins and glycoproteins present in the plasma. These are mostly synthesized by liver hepatocytes. Okay, and some of the components are synthesized by other cell types, that is in smaller proportion. The rest major one is secreted by the liver hepatocytes. Okay, and it constitutes about 5% of the serum globulin proteins. They circulate as inactive prozymes or zymogens. Okay, so they constitutively circulate in the body or in the plasma, and these are basically proenzymes. That means they are in the inactive form. Okay, so once they are activated, they can now activate the cascade after the activation. And their proteolytic cleavage removes inhibitory fragment and exposes the active site. So this is how they gets activated, these proteins. And what is their main function? Their main function is lysis. Okay, lysis by forming the membrane attack complex. Opsonization basically means that you are coating certain proteins on the microbes or the cancer cells, right? So after coating these particular proteins, they can be actually identified by the immune system and they can be cleared off. Okay, so these proteins that are used for coating are called as opsonins. Okay, so for example, there is a bacteria which was not recognized by the immune system, but after coating with the complement proteins, what happens? This was actually being this was identified by the phagocytes, and now the phagocyte will engulf this and will drive to the adaptive immunity. Okay, and the third function is the activation of the inflammatory response. So, what will happen now? The complement proteins basically are the ligands or the receptors. Okay, so they lead to degranulation of the granules present in basically agranulocytes. Okay, so that leads to the release of histamine and serotonin. So, this leads to inflammation. So these are the three major functions of the complement system: lysis, opsonization, and the activation of the inflammatory response. Okay, so any doubts in this particular slide? Okay, if no doubt, then we'll move on to the next slide. So now there are certain proteins that are involved in the complement pathway. Okay, so first we'll check for their naming, how they are named. So Uh, they are basically either designated by numerals, for example, C1 to C9, the letter symbols like letter factor D, or the trivial names, for example, homologous restriction factor or MBL, mannose binding, mannose binding lectin, like that. Okay, so there are three kinds: either numerals, letter symbols, or trivial names. The peptide fragments made by the activation. So as these are inactive zymogens, so whenever they are activated, they produce two kind of peptides. Okay, so one is the A, that is the smaller fragment, and B, that is the larger fragment. Okay, so either of them can function in different way. So that's why both of them have a different function, and both of them can be used in activating either of the immune components. Okay, and if there is enzyme active, if the complex have an enzymatic activity, so they are represented by a bar. So for example, the C4B2A is actually functioning as an enzyme. That's why the whole complex is supported by a bar. So this is how we name them. Now we'll move on to the different proteins that are involved in the complement pathway. So for there are seven classes of proteins that are present. Okay, so the first is initiators. So initiators are basically those proteins that initiate the complement pathway. Okay, so there are three different types or the classes of pathway that we've studied in the further slides. And so because there must be something that actually attach attach to the ligand, and it directly initiates the complement pathway. So those are called as initiators. For example, we have the C1q, MBL, or ficolins. So these three basically help in initiating the complement pathway. Now, after initiation, there are convertase activators and enzymatic mediators. So that leads to the pathway for to actually amplify. Okay, so the amplification phase takes place after this. So these are the convertase activators. So example are C1R, C1S, C4B, and C2A. So these are the convertase activators, and the enzymatic mediators like C3 convertase and the C5 convertase are the enzymatic mediators that help in converting the inactive form to the active form. Okay, now what they'll do, they'll actually activate the opsonins. So this process is known as opsonization. So the third class comes in opsonins. So opsonins are basically, as I discussed, the proteins that coat the bacteria for their recognition by the antibodies or the phagocytes to help in clearance of those particular organisms. Okay, so these are the opsonins. Then anaphylotoxins that leads to inflammation. Okay, so there are certain proteins. This is the fourth class, that is anaphylotoxins that leads to the inflammatory response or the release of histamine. Okay, then we have the other class, fifth one, that is the membrane attack complex. So these proteins form a pore on in the membrane. Okay, so they form a pore. So these classes of proteins actually help in forming the pore around the bacterial or any microbial membrane, so that they have the osmotic disruption and the cleavage of their cells. Okay, so this is the membrane attack complex, and then we have the complement receptors, that is the sixth class. So the complement receptors basically help in activating the functions of complement. Okay, and then we have the regulatory proteins, the seventh class, that actually degrade those components when they are not needed. Okay, so this comes to the seventh class, that is the regulatory components or the regulatory proteins in the uh, complement pathway. Okay, so here are the seven classes of proteins. So all the complement system will revolve around these seven classes of proteins. And now we'll move forward to see how these complement pathways actually function. Okay, so any doubt still here?
Uh, Good evening, sir.
Good evening.
Uh, Sir, actually, in example, it's showing complement protein from 1 to 9, but you are saying it's total complement protein, total like 7 classes. So I'm little bit confused in there.
Okay, so the total number of proteins are divided into C1 to C9, but those C1 to C9 are divided into seven different classes. So they have a different function that can be distributed either in of the seven classes.
Okay, sir.
Okay, so for example, as you can see here in the second, that is the convertase activator and the enzymatic mediators, there are different classes like C1R, C1S, C4B. So all of
Yeah.
these proteins will be divided into seven different classes.
Okay, sir.
Okay, so let's move forward. Now, there are majorly three pathways through which the complement system activates. Okay, so first is the lectin pathway. So the lectin pathway starts with the mannose binding lectin and the ficolins. Okay, so these two proteins basically recognize the carbohydrates on the pathogen surface. Okay, so the bacteria, for example, have certain carbohydrate moieties on their surface. Okay, so these carbohydrate moieties are basically identified by mannose binding lectin or ficolins that activates this lectin pathway. Okay, as you can see in the figure also, so this is a ficolin and this is a mannose binding lectin. Now this so we have MBL ficolin, MBL or ficolin, then MASP2, C4 and C2. So these are majorly involved in the lectin pathway. So these are the complement proteins that are involved in the lectin pathway. Now moving on to the classical pathway. So classical pathway is basically starts with the antibodies. Okay, so this classical pathway is considered to be a part of the adaptive immunity because when antibodies recognize the pathogens, then only this C1q, that is the major initiator protein in the classical pathway, will start acting and it will now replicate, it will activate the further proteins. Okay, so when antibody binds to the surface, C1q interacts with the antibodies and then it activates different proteins. Okay, as you can see here, so this is an antibody that is bound to the surface. Now this C1q and C1s will now activate this because of this antibody. Okay, and here are the proteins that are majorly involved, that is C1q, C1r, C1s, C4 and C2. Okay, so these are the proteins that are involved in the classical pathway. Now moving on to the third pathway, that is the alternative pathway. Now in alternative pathway, this pathway is termed as antibody independent pathway. Okay, so now the C3, upon spontaneous hydrolysis, initiate the deposition of C3 convertase on microbial surfaces. Okay, so whenever the C3 is basically hydrolyzed, that means addition of H2O, this initiates the deposition of C3 convertase on the microbial surface, then the alternative pathway actually initiates. Okay, and there is another factor, that is propyridine or the factor P, that also helps in the initiation of the alternative pathway. Okay, so and here the factor D, factor B, propyridine, and C3 are the major proteins that are involved in the alternative pathway. So there are different set of proteins that are in majorly involved in these three pathways. Okay, so why these three pathways came in? So because the complement system need a ligand to basically start the process. So these three pathways basically describes how a complement system or a complement pathway activates. So these three pathway helps us understand that there can be different mode of activation of the complement pathways. Now there is second phase that is called as the amplification phase of the complement, and this amplification phase is common to all the pathways, and it starts with a C3 convertase, which so a C3 convertase is activated, which cleaves the C3, okay, leaving the C3b bound to the microbial surface and releasing C3a. So C3 is converted to C3a, that is the smaller domain, and C3b the larger domain. Okay, C3b the larger domain is bound to the micro cell surface, and C3a is uh, like detached from C3b, and now it can activate different inflammatory components. Okay, so this C3 pathway is common to all the three pathways. Now there are three like functions that takes place after the complement is activated. Okay, so for example, there is recruitment of the phagocytic cells. First thing, there is the engulfing of the opsonization, because there were certain proteins that are bound to the surface of the microbes, and the third one is the formation of the membrane attack complex. Okay, so for example, C3a and C5a, okay, the smaller components of the C3 and C5, they recruit phagocytic cells to the site of infection and promote inflammation. Okay, so one function is the recruitment of the phagocytic cells by the C3A and C5A. The second is opsonization. So, phagocytes with receptors for C3b engulf and destroy the pathogen. So, C3b, as we saw here, was attached to the microbe itself on the surface. Now, that actually helped, that acted as a opsonin. Okay, so those opsonins helps in opsonization of the microbe. And the third one, after the completion of the complement cascade, leads to formation of a membrane attack complex that disrupts the cell membrane and causes the cell lysis. Okay, so there are multiple components, as you can see here: C5b, C6, C8, C7, and C9, that helps in the formation of a membrane attack complex so that cell lysis can take place. Okay, so this is how a complement pathway, all the three pathways, how they start, how they merge, and what they function. Okay, so this was a brief. Now we'll get into the details of all the three. Okay, so any doubts in this particular slide? Okay, if not, then we'll proceed. So this is the detail of the complement pathways. Okay, now as you can see here, the classical pathway start by the antibody, then the C1q and other proteins are involved, and it activates the C3 convertase, that is this one. Okay, the lectin pathway starts with MBL, then the cell wall, and how likewise it activates the C3 convertase. And the alternative pathway directly starts with C3 and it activates the C3 convertase. Okay, so all the three pathways have different initiation points, but they merge on the C3 convertase, and then they form the membrane attack complex. Okay, now we'll see each pathway in detail, starting from the classical pathway. So the classical pathway is termed as antibody dependent. That's why some of the literature suggests that it is part of the adaptive immunity. Okay. So when there is a bacteria that shows certain epitopes. Okay, so these epitopes are bound by the antibodies. So these antibodies now activate the complement system. Okay, so these are activated by the antibodies bound to the epitopes on the microbes. Now, there are two types. So this pathway is actually activated by two kinds of the pathway, that is soluble antigen antibody complexes and the immune complexes, basically, that are bound to the microbes. Okay, so both soluble as well as membrane bound antibody can activate the classical pathway, and mostly IgM and some classes of IgG are involved in the activation of the classical pathway. Okay, so and the early stages involve C1, C2, C3, and C4. So these four proteins are involved in the classical pathway. Okay, so now we'll see the C1 macromolecule and what is its structure. Okay, so the C1 macromolecule, as we see here, is a complex of C1q, R2, and S2. Okay, so as you can see here, this is the C1q. These small proteins are R2. So the C1R, there are two molecules, and C1S also two molecules. Okay, so this is how it is represented. So C1q has a stalk. Okay, so this has a collagen like triple helix that forms a rigid rod like structure of this particular C1q, and it has heads. Okay, now this C1q basically has in like three molecules. So one C1q has two heads. So into three, that means there would be six heads. Okay, so there are six heads of the C1q, and then we have one C1, two C1R, and two S2. So that's why the nomenclature is like this. Okay, so this is a macromolecule of the C1q. As you can see here, this is the electron micrograph of the whole macromolecule. Okay, so now this molecule helps in binding to the antibody that is bound on the microbial surface. Okay, so this is how it binds because of its structure. Now this is the pathway. So first what happens, as you can see here, this is the antibody. So antibody has two regions. Okay, so basically we have the heavy chain, the light chain. Okay, so the light chain is only single chain and the heavy one is dual chain. Okay, and then it has a FC region. So FC region is the crystallized part, or can be crystallized part of the antibody. That's why it is known as FC region. And FC region helps in attaching the C1q. Okay, so it acts as a receptor for the C1q. So this antibody is bound to the particular microbe. Now it recruits the C1q. Okay, the C1q that have R2 and S2. Now, what happens? The C1R activates autocatically. So the R2 or the R domain has a catalytic domain. That means it has a active site and it can auto activate itself also. Okay, so it auto activates itself and the second R2 and the second R unit, and both of them activate the S. So R is active, and then it activates itself, the other R unit, and both the R units, when they are activated, they activate the S units. Okay, now what will happen? The S units, after activating, they attract C4 and C2, the two more proteins of the complement system. Now, whenever they are activated, so they recruit C4 and C2 and they cleave them. So now C4 and C2 will be divided into C4A and C4B. Okay, so this is the C4A and the C4B is here. The C2 is also converted to C2B and C2A. Okay, now C4, after cleaving the C4, it exposes the binding site for C2, and C4 binds to the surface near C1 and C2 binds C4, forming C3 convertase. Okay, so what will happen whenever this thing happens? The C4 is the site to which the C2 binds is exposed, and this leads to the binding of C4B and C2A. So C4B and C2A, after combination, forms the C3 convertase. Okay, so that leads to the activation of the C3. So this is the C3 convertase enzyme that is formed by the by the action of C1s on C4 and C2. Okay, so this was the second step. The third step is the C3 convertase now hydrolyzes C3 molecules into C3b and C3a. Now, and then what will happen? The 3b. Okay, so the 3b, that is the larger fragment of 3, attaches to the C4b and 2a, that is the C3 convertase, and activates a C5 convertase. Okay, and the C3A, this will attract the phagocytes for the inflammatory response. Okay, now the C3B component of the C5 convertase binds to the C5, and now it will cleave C5 into C5B and C5A. Now C5A will lead to the inflammatory response or the recruitment of the phagocytes, and C5B now will help in formation of the membrane attack complex. Okay, now this C5B binds to C6. So C5b binds to C6 and initiates the formation of the membrane attack complex. Okay, so now it will recruit C5, C6, then 7, then 8. So as you can see here, this is a rod-like structure. Okay, now after attaching this, the there is conformational change in 8 that recruits the C9. Okay, so C9 is basically rod-like proteins that helps in initiation of the formation of the pore-like protein on the microbial surface. Okay, so after this pore is formed, so this forms the membrane attack complex. Okay, so this is basically C5b, C6, 7, 8, and poly 9. So this is the membrane attack complex that are formed by the activation of the C5b. Okay, so this was the whole classical pathway that was being told just now. So any doubts in this particular pathway? So to brief about this, this was activated by the antibody. So the first is antibody that binds to C1q. C1q has two components, R2 and S2. R2 activates itself, the other R domain that activates S. So S recruits C4 and C2, cleaves it. So C4b and C2a form the C3 convertase. C3 convertase cleaves C3, that leads to the formation of C5 convertase, that is attaching C4b, C2a and C3b, and C3a leads to the recruitment of the phagocytes. Then C5 convertase cleaves C5 into C5b and C5a. C5b basically recruits C6, then C7 and C8, and then after combining these three, it recruits C9. Okay, so this helps in formation of the poke inside the membrane, and then recruits the C9, and the poly C9, when they are recruited, these form the membrane attack complex. Okay, so now moving on to the second pathway, that is the alternative pathway. Now, this pathway is termed as antibody independent pathway because it is not initiated by the antibody, and it is considered to be a part of the innate immune system by some literature. Okay, the classical pathway was a part of the adaptive immune system, but the alternative pathway is part of the innate immune system. And here involves the C3, factor B, factor D, and properdin. Okay, and these are initiated by cell surface constituents foreign to the host, and these are the gram negative and gram positive bacteria that initiates help in initiating this pathway. Okay, so the pathogens and particles of microbial origin, for example, gram negative bacterial strains, lipopolysaccharides from gram negative bacteria, gram positive bacteria, then teichoic acid from gram positive cell walls, fungal and yeast cells, that is zymosan, some viruses and virus infected cells, tumor cells, and parasites. So these all components lead to the activation of the alternative complementation pathway. Okay, so now we'll see how it gets activated. So as you can see here, the important component is C3. So now what happens is C3, when it is hydrolyzed, it is termed as C3, that is in bracket H2O. It recruits factor B, another protein that is involved in the complement pathway. After attaching to factor B, it recruits factor D that cleaves it into a small and a large domain, that is BA and BB. Okay, now what will happen? C3 and the larger component of B, that is BB. So this forms the C3 convertase. Okay, now this C3 convertase will cleave C3 into C3 and C3b. Now C3bb will, so now C3b and bb will form the membrane bound C3 convertase. Okay, so this is the completion of the formation of the C3 convertase, that is membrane bound. Now the sorry, this C3 convertase that was formed here, this C3 convertase now bound to properdin, that is a factor P. So now it will convert C3 to C3a and b, which will form the C5 convertase. Now C5 convertase will lead to the activation of the MAC, that is the membrane attack complex. Okay, so here are different components involved that are activated based on the different initiation points or the components of the different bacteria. Okay, so this can be activated by any stimulus that is mentioned here. That's why it is a part of the innate immune system. So it can be activated by any of these stimuluses. Okay, so this is the case with the alternative pathway. So any doubts in this pathway? Okay, if not, then we'll move to the third pathway, that is the lectin pathway. Okay, so as you saw here also, in the alternative pathway, although it starts with C3, now it leads to the formation of a membrane bound C3 convertase that forms either forms C5 convertase, and then C5 convertase leads to the formation of the membrane attack complex. Okay, so this C3 convertase is the connecting link between all the three pathways. Now, the lectin pathway is also antibody independent, and it is initiated by the mannose binding lectin to binding to of this MBL to the mannose residues on the glycoproteins. Okay, so the N-linked glycoproteins of yeast contain many terminal mannose residues, whereas glycoproteins of vertebrates have terminal sialic acid residues. Okay, so as you can see here, these green colored circle boxes are the mannose. Okay, so this yeast proteins have many terminal mannose proteins as compared to the vertebrates. So these are recognized by the MBL that is present in our body and in many bacteria. For example, here also we can see the bacteria that have the mannose that is bound on its surface leads to the activation of the lectin pathway. And here are some of the causative bacteria and the viruses, some fungal cells also. So this microorganisms that have mannose starts the lectin pathway. Okay, so this lectin pathway starts with the mannose binding lectin on the microbial cell wall. Now, after binding of this mannose binding lectin, the MBL associated serine proteases gets activated. Okay, so this serine proteases activates the C1 like complex. Okay, so likewise, this was the C1Q. So this C1 like complex was activated. So we have the MBL lectin as the C1Q. Then MASP1 and serine protease 1 and 2 are C1R and S, like in the classical pathway of the complement system. And then this leads to the formation of the conversion of C4 to C4b and C4a, and C2 same to C2a and C2b. So for the formation of the C3 convertase, then formation of the C5 convertase, and then formation of the membrane attack complex. Okay, so this lectin pathway basically is initiated by a sugar moiety with the specific proteins. But these proteins are analogous to the C1 complement protein that is present or the part of the classical pathway. Okay, so any doubts in the last two pathways or till here? Okay, let's move forward. So the final key point is the membrane attack complex, formation of the membrane attack complex that will lyse the microbial cell by osmotic imbalance, and that will lead to help in the formation of the pores. So this membrane attack complex is formed by C5b that recruits C6. After binding, it recruits C7, and after its binding, it recruits C8. So it has the affinity of C8 now. So the C8 helps in piercing the membrane first. After its recruitment, it pierces the membrane first. Now, after binding to C7, its conformational changes. Then it leads to the recruitment of C9. Okay, so now this C9 also starts piercing the wall or the membrane, and multiple C9 moieties actually forms a complex that leads to the formation of pore in the membrane. Okay, as you can see here, so this is a ring-like structure that was seen in the electron micrograph, or to that leads to the membrane attack complex. Okay, so this is how a membrane attack complex is formed in a particular system, and this is the most important pathway how a microbe is actually lysed inside the cellular body. Okay, and this membrane attack complex helped Bordet. So remember, he actually did some of the experiments that in on Vibrio cholerae that it was lysed from the antiserum. So this lysis basically takes by the formation of the membrane attack complex. Okay, so moving forward, so if there are any doubts, you can ask till now. So this was the complete pathway how a complement basically functions. Okay, let's move forward. So sorry. Now, because these pathways are part of recognizing and clearing of the pathogens. Okay, so these pathways actually help. So these pathways require some regulation, because if these pathways get activated constitutively, that will lead to the harming of our self cells also. Okay, for that, regulation is needed for these complement pathways. Now, complement components are capable of attacking the host cells. Okay, that's why its regulation is needed, and they undergo spontaneous inactivation if they are not stabilized with other components as it is highly labile compounds. Okay, so these are highly sensitive compounds. That's why spontaneous inactivation is needed. Okay, then the C3 convertase is the major amplification step in all the three pathways that need the regulation, because C3 convertase is a common pathway of the three pathways. Now, this C3 convertase, if it gets constitutively active somehow, then it will actually lead to the harm to the body cells itself. Okay, so that's why the regulation is needed in this function also. So now we have certain proteins that are part of the regulatory component of the complement system. So remember class 7 that have the regulatory proteins. Okay, so these are the regulatory proteins that are involved in the complement system. Now, the first one is C1 inhibitor that is present in a soluble state in the plasma, and it affects the classical and the lectin pathway. It induces the dissociation and inhibition of R2S2 from C1q, that is, it is a serine protease inhibitor. Okay, so you remember C1q has just 1 minute. So you remember C1 has different components. So C1q has C1R2 and S2 also. So R2 and S2 are serine proteases that activate C4 and C2. Now, to inhibit these components, the serine protease, we have the C1 inhibitor, that is C1NH. So this inhibits this binding of the C1q with the R2 and S2. So it dissociates C1q with R2S2 so that it remains in the inactive state. Once it is activated, it binds to the C1q, has a conformational change, and then activates itself S2 and further the C4 and C2. Okay, so the first protein is C1 inhibitor. Second is decay accelerating factor, or the DAF, CD55. So this is a membrane bound protein that affects all the three pathways, and it accelerates dissociation of C4b and C2a and C3b and BB. That is also a C3 convert. That is the C3 convertase of the alternative pathway. So it regulates the C3 convertase. Then we have CR1, that is CD35, also a membrane bound, affects all the three pathways. It blocks formation of or accelerates dissociation of the C3 convertase by binding C4b or C3b. Okay, so it also affects the C3 convertase by binding to C4b or C3b and helps in dissociation of its components. And it is a cofactor for factor I in C3b and C4b degradation on host cell surface. So these two are involved in the regulation of the C3 convertase. Then we have C4BP, that is soluble, and classical and lectin pathway are its targets. This blocks the formation or accelerates the dissociation of C4b to a C3 convertase, and it is a cofactor for factor I in C4b degradation. So this also is a part of the C3 convertase regulation. Then we have factor H. This is also soluble, affects only the alternative pathway. It blocks the formation of the C3 convertase by helps in degradation with the C3b degradation. Okay. Then factor I. This is also soluble and it can affect all the three pathways. It is a serine protease that cleaves C4b and C3b, that will actually lead to the degradation of the C3 convertase. Okay. Then we have membrane cofactor of proteolysis, that is MCP CD46. So this is a membrane bound complex, and this is affects all the three pathways. It is a cofactor for factor I that helps in degradation of C3b and C4b. So another factor that will regulate the C3 convertase. Then we have S protein or vibronectin, vitronectin. So vitronectin is a soluble protein that affects all the pathways. It binds to C5b67 and prevents insertion into the host cell membrane. So now here we have the regulation of the membrane attack complex. Okay, so the S protein will inhibit the association of C5b6 and 7, and it will prevent its insertion into the host cell membrane. Then we have protectin, that is CD59, that is also membrane bound, and it affects all the pathways, and it binds to the C5b678 complex on the host cell, blocking binding of C9 and formation of the membrane attack complex. Okay, now we have the carboxypeptidase NBRR. So these are soluble peptidases, and these are anaphylotoxins produced by all the pathways. They help in cleaving and inactivating the anaphylotoxin C3 and C5A. So we have only one inhibitor of C1. We have six inhibitors of the C3 convertase, two inhibitors of the formation of the membrane attack complex, and one inhibitor for the anaphylotoxins, that is C3 and C5A. Okay, so this is how the complement system is regulated inside the human body. Okay, so any doubts in this particular question? Okay, we'll move further. So this is how the different proteins actually work upon. Okay, so just now we discussed. So this is a diagrammatic representation basically. So first is C1NH. So this dissociates R2 and S2 from C1Q so that they will not be activated. Then we have C3 convertase decay accelerating activity, that is DAF, CR1, C4BP, and for the alternative pathway, we have DAF, CR1, and factor H that will basically dissociate C4B and C2A and C3B and BB. Okay, then we have factor I cofactor activity. So this factor I also dissociates the C3 convertase, uh, degrades the C4 and the C3 components of the C3 convertase. Then the MAC inhibition. So we have protectin and vitronectin. So these prevent the formation of the membrane attack complex. And then we have the anaphylotoxins. So carboxypeptidases N, B, and R, they cleave C3A and C5A to lead to their cleavage and the inhibition of the inflammatory response. Okay, so this was the basically diagrammatic representation of what we have studied in the last slide. Okay, now there are certain diseases that are associated with the complement pathway. Okay, so for example, how there because it is a pathway and microbes are intelligent than us, so they evolve evasion strategies so that they can evade the complement system, and they can actually infect the human body. Okay, so some of them have been studied. For example, Neisseria, Borrelia, and Streptococcus have certain membrane proteins that resembles factor H. Okay, so this factor H helps in inactivation of the bound C3b from the alternative pathway. Okay, so this bound C3b is inactivated, so that means the C3 convertase is inactivated. So it will not be able to form the membrane attack complex. So the major amplification step is inactivated. Okay. Now we have certain secreted proteins like from Neisseria, Staphylococcus. So there are certain secretory proteins like core A, clumping factor A, then Staphylococcus protein A, staphylokinase, and complement inhibitor. Okay, so these are the proteins that are produced by the bacteria or the fungi, and these affect C4BP, factor I, immunoglobulin, that is the antibodies, and the C3 convertase. So it inactivates the C3b and inactivates the convertase activity, and the staphylococcus protein and staphylokinase, they cleaves the immunoglobulins and bound to the FC region so that they will not be able to recruit the C1 protein. Okay, so this is how certain bacteria have evolved strategies to evade the immune system or the complement pathway. Okay, now deficiency of either of these complement proteins show enhanced immune complex and show SLE like symptoms. Okay, glomerulonephritis and vasculitis. Individual also show recurrent infection by pyogenic, that is pus forming bacteria. So if there is deficiency of either of these complement proteins, will show enhanced immune complex formation. That means the formation of the membrane attack complex, and then these can lead to certain diseases. Deficiency of factor D and properidin show Neisseria meningitis infection but not with meningococcal bacteria. Deficiency with MBL, that is mannose binding lectin, shows in respiratory tract infection because the bacteria that cause the respiratory tract infection are mostly have mannose in their membrane. Okay. Deficiency of C3 shows frequent severe bacterial infection. Protectin inhibits C9 before, therefore MAC deficiency has high risk of thrombosis. Okay, then that means coagulation of blood. Anti C5 is used for the treatment of paroxysmal nocturnal hemo, hemoglobinuriria (PNH) and is come in the name of eculizumab. Okay. Then we have deficiency of C1 LNH cause hereditary angioedema. So these are the diseases that are caused by either the deficiency or the increase in formation or increase in the bacterial infections and some of the diseases. Okay. So any doubts still for from the regulation of the complement system? Okay, if not, then we'll start with the questions that are associated with the complement system. Okay, so select all the statements regarding the C3 convertase in the alternate classical and the lectin pathways. So, what can be the correct answer? 2, 3, 4, and 5. Okay, so let's see the options. So C3 convertase in the alternative pathway consists of C4b2A. No. Okay, for the alternative pathway, the C3 was C3b and BB. Okay, so this was the convertase in the alternative pathway. So the first option is false. Then properidine stabilizes the C3 convertase in the alternative pathway. Yes, this is correct. In the classical pathway, C3 convertase is formed by C4b2A. So in the classical pathway, C3 convertase is formed by the C4b2A. Yes, this is correct. C3 convertase activity leads to the generation of C5 convertase. Yes, this is also correct. So when the C4b2A is formed, it cleaves the C5. So C5, then addition with the 5b, it leads to the formation of C5 convertase. Okay, then the lectin pathways C3 convertase is identical to the classical pathways. Yes, not identical, but rather it would be similar. Okay, no, it is identical because only the C1 has a difference. It activates C4 and C2. So yes, it is identical. Okay, then the mannose binding serine proteases 2 is essential for C3 convertase formation in the alternative pathway. No, it is involved in the lectin pathway. Okay, the C3 convertase is stabilized by C3b in the classical pathway. No, in the alternative pathway. C3A generated by the C3 convertase acts as an opsonin. No, it acts as an anaphylatoxin that activates the phagocytes. Okay, so hence option 2, 3, 4, and 5 are correct. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. What is the significance of enzyme C5 convertase in the classical complement pathway? So, what can be the correct answer? Okay. Yes, the correct answer is option 1. So, C5 convertase actually leads to the C5b leads to the recruitment of C6, 7 and 8 and eventually 9. So, these forms the membrane attack complex. Okay. Now, moving on to the next question. Which antibody type is most effective in initiating the classical complement pathway? So, what can be the correct answer? So, yes, IgM is the correct answer because IgM has is a pentameric antibody. Okay, so what will happen? It can easily bind to any of the epitopes on the microbes, and it can activate the complement pathway. IgG, on the other hand, is only dimeric in nature. Okay, so it will need more number of IgG molecules to actually initiate the classical pathway. That's why IgM is more effective here. Okay, at IgM, has specificity towards many organisms. Okay, so that's why IgM is part of the classical complement pathway. Okay, let's move on to the next question. Which disease is primarily associated with a deficiency in C1 inhibitor? So, what can be the correct answer? So just now we see a couple of slides back. So this is hereditary angioedema. So the deficiency of C1 inhibitor, that is C1NH, that leads to hereditary angioedema, but not SLE. Okay. So deficiency of C1, C4, C2, that leads to SLE, that is systemic lupus erythematosus. Okay. So let's move on to the next question. Why is IgM considered more potent than IgG in activating the complement system? So what can be the correct answer? Just a few questions back we discussed that IgM molecule is pentameric. Okay, so it can directly activate the complement system, whereas multiple IgG molecules will be required to activate the C1q, that is the classical complement pathway. So the correct answer is option number 3. Okay. So let's move on to the next question. What is the role of C3 in the alternate and the lectin pathways? So what can be the correct answer? Yes, someone has raised the hand. Okay. So the C3 in the alternate and the lectin pathway helps in the formation of C3 convertase. Okay, so this C3 convertase will now lead to the amplification of the complement pathway. Okay, so the correct answer is option number 2. So any doubts in this particular question? Okay, so let's move on. What is the key benefit of the alternative pathways amplification loop? So what can be the correct answer? Okay. So yes, the correct answer is option number three. So in the alternative pathway, the three C3b actually attaches to the membrane. Okay, so this C3b when attached to the membrane will act as an opsonin. So this opsonin is basically coating of the C3b to the micro, and that will be detected by the phagocytes. Okay, so yes, the third option is correct. Okay, so any doubts in this particular question? Okay, if not, then we'll move on. Which of the following steps are involved in the activation of the classical complement pathway? So, what can be the correct answer? Okay, 1, 2, 3, 5, 7. Okay, any more answers? Okay, 1, 2, 3, 5, 7, and 8. Okay, so let's see the answer. So we have one. that is binding of IgM, because the classical pathway starts with the antibodies. So the IgM or IgG antibodies to the antigen binding. Yes, this is the path. Then we have C1q binding to the CH2 domain of the antibodies. Yes, this is also correct. Okay. Then we have the activation of C1R and C1s through proteolytic cleavage. This is also correct. Then we have direct cleavage of C5 into C5 and C5b. No, this was cleaved by the C5 convertase. Then the formation of C3 convertase, yes, C4b and 2a. Then inhibition of the complement activation by factor H. No, factor H is involved in the alternative pathway. Okay, then the formation of C5b convertase, sorry, C5 convertase, then recruitment of C4b and C2a to the antigen antibody complex. Yes, this is also correct. Although the C4 and C2 are recruited by the activation of C1s. Okay, then they are cleaved. So it can be correct. Okay, so the correct answer would be 1, 2, 3, then 5, 7, and 8. Okay. Any doubts in this particular question? Okay, so let's move on to the next question. Which of the following statements are correct? So, what can be the correct answer? Okay, 3, 2, 7. Okay, any more answers? 1, 3, 4, and 5. 3, 4, and 5. Okay, so let's see. The complement components are capable of attacking host cells also. Yes, this is correct. Then C3 convertase is not regulated in all three types of complement pathways. No, this is false. It is always regulated. That's why it will not attack its own cells. Complement components are highly labile. Yes, this is correct. Then we have deficiency of C1 inhibitor, hereditary angioedema. Yes, this is also correct. Deficiency of C3 shows frequent severe bacterial infections. Yes, this is also correct because C3 is the major part which helps in the formation of C5 convertase and the membrane attack complex. So, whenever there will be deficiency of C3, will lead to more severe bacterial infections. Deficiency of MBL shows an increase in the respiratory tract infection. Yes, because the respiratory tract infection caused by the bacteria that have manners. Soluble proteins and glycoproteins are synthesized mainly by the hepatocytes. Yes, this is also correct. Classical complement pathways antibody independent. No, they are antibody dependent. Lectin complement pathway is antibody dependent. No, they are antibody independent. Okay, so the correct answer is option 1, 3, 4, 5, 6, and 7. Okay, so any doubts in this particular question? Hmm. Okay, so now we'll be moving on to the first we covered with the innate immunity, the components, the receptors, the recognition patterns, and how now we'll move on to the adaptive immunity. Okay, so the adaptive immunity is more specific in nature. Okay, so as we see here, so there are cell components that are associated with the innate immunity that can be both immune and non-immune cells. So they recognize PRRs, they have pattern recognition receptors that recognize pathogen associated molecular patterns or the PAMs. Now they initiate inflammation, induce interferon release, and activate innate immune responses. Now, after all this, there will be activation of the specific immune response via the B and T cells. So, immune response is known as adaptive immune response. Okay, so this adaptive immune response is majorly taken care of by the B and the T lymphocytes. Okay, so there are immune cells that are neutrophils, basophils, eosinophils, and the NK cells that are involved in the innate immune system. Now, these actually present the antigen to the macrophages and the dendritic cells that are the phagocytes involved in multiple pathways. Okay, so these phagocytes now link the pathogen to the B and the T lymphocytes, and they'll increase their immune response by the adaptive immune cells. Okay, so now we'll discuss different properties of the adaptive immune system by comparing it with the innate immune system. Okay, so starting with the presence, that means the innate immunity is already present in the body, but the adaptive immunity is not present by birth. Okay, so it develops upon exposure to the foreign antigens or the foreign substances. Then specificity, innate immunity has a broad specificity, but the adaptive immunity is highly specific. So the adaptive immunity will be developed only for a particular pathogen that is encountered by the body. But the innate immunity is developed by for any of the organism that is present or can infect the body. Okay. The response, it fights with any living and non-living foreign entities. It develops highly specific immune response if not cleared by the innate immunity. Okay, so the response is created by the adaptive immunity. The time for which the innate immunity functions is rapid. Okay, so it's from minutes to hours because it is the first line of defense of the body. So it must be rapid. Now, in the adaptive immunity, it is slow. So, because after the innate immunity is not able to properly overcome the infection, now it leads to the adaptive immunity and it's slow. It takes around days or weeks to start the immune response. Okay, then for potency, it is low, limited and lower potency, and the adaptive immunity as a high potency. Okay, the time span, every time the response is generated, irrespective to the number of exposure. Once the immune response is developed, the immunity can be lifelong or short depending upon the pathogen. Okay, so this is the comparison with the innate and adaptive immunity. Now, inheritance, the innate immunity is not inherited, sorry, is inherited from the parents to the offspring. Okay, so there is a vertical inheritance pattern in terms of the innate immunity, but for the adaptive immunity it is not passed. Okay, so that means it is not inherited from parents to offsprings. The intensity of the immune response is, for the innate immunity, it depends upon the exposure of the pathogen. Okay, if the pathogen amount is more, that means the innate immune response would be intensity would be more as compared to when the exposure was small. In case of the adaptive immune system, the specific pathogens, because they have some memory of that particular pathogen. So what will happen if there is the pathogen is encountered again, that means the adaptive immune system can develop an immunity that is very much increased. Okay, then presence. So the presence is present at the time of birth and it is developed when the person starts growing, and it can be short lived. Okay, the allergic reaction. Yes, there are in innate immunity there are no allergic reactions associated, but in the adaptive immunity there can be, and there can be immediate or delayed hypersensitivity. In terms of memory, the innate immunity develops no memory, but the adaptive immunity has long term memory. If you would have encountered any of the microbes in your childhood, will develop immunity, and if you encounter it after some time also, then the immunity will be developed and you will be saved by from that infection. Okay, then for the diversity of the innate immune system is limited, while the adaptive immune system is high. Okay, the complement system activation in terms of the innate immunity is the alternate and the lectin pathway, and adaptive immunity is the classical pathway because of the involvement of the antibodies. Then the anatomic and physiological barriers include skin, mucous membranes, temperature, pH, chemicals, etc. for the innate immune system. And for the adaptive immunity, we have the lymph nodes, spleen, malt, and if talking about the cells, we have the antibodies and T and B cells. Okay, composition. It is composed of physical and chemical barriers, phagocytic leukocytes, dendritic cells, NK cells, and the plasma proteins, the complement proteins. For the adaptive immune system, we have the B and T cells. So B cells basically secrete the antibodies. Okay, the development, evolutionary and older, and is found in both vertebrates and invertebrates, while the adaptive immune system is only recently found in vertebrates only. They are not found in the invertebrates. For example, the WBC fighting bacteria causing redness and swelling when you have a cut. So this is an example of the innate immunity. And then the vaccination process, for example, chickenpox vaccination, so that the chickenpox will not be we will not get the chickenpox because of the adaptive immune system. The body has remembered those chickenpox vaccines. Okay, so these were some of the properties of both innate and adaptive immune system. So this would help you guys to actually differentiate between the two, and how adaptive immune system is important as compared to the innate immune system. Okay, so the B and the T cells are the major cells that are involved in the adaptive immune system. So the B cells, they basically produce the antibodies, and T cells, they produce cytokines so that they can activate different uh, phagocytes for the clearance of the uh, microbes. Okay, so interaction with the antigen involved by either the immunoglobulin and the antigen. Okay, and it is with the T cell receptor, antigen, and the MHC molecule. So this is the major histocompatibility molecule. So this we'll study in the next few chapters, how a T cell is actually presented an antigen from a foreign as well as the self moiety. Okay, then binding of the soluble antigen. Yes, B cells can directly bind through antibodies to the soluble antigen, but the T cells cannot bind to any of the soluble antigens. Okay, involvement of the MHC molecule. B cells do not require MHC molecule, but T cells require these MHC molecules to present the antigens to the T cell receptor. Okay. Chemical nature of the antigens, the protein, polysaccharide, lipids can be presented to B cells, but mostly proteins are involved in the T cells. Some of the lipids have been seen to be presented to T cells, and on the MHC like molecules. Okay, so the epitope property. So epitope means any molecule that is present on the surface that is bound by the antibody. Okay, so there can be multiple epitopes that are present on the surface that can be bound to particular antibody. Okay, so these epitopes of the B cells can be accessible, hydrophilic, mobile peptides containing sequential or non-sequential amino acids. While for the T cell, there are linear peptides. Okay, that have been processed during the MHC presentation that are bound to the MHC molecules. Okay, so these are the differences between the B and T cells, how they function. Okay, so these we'll see in detail in a couple of slides in the next few weeks. Now, moving on to the important concept of the antigen. Okay, so we just now discussed about the epitopes. Now, we have the two terms, that is antigen or the immunogen. Okay, so antigen is one that is capable of binding specifically to a T cell receptor or an antibody. Okay, so any molecule that can bind to the T cell receptor or antibody is termed as an antigen. Okay, and the ability of binding to these particular two molecules is known as antigenicity. Then we have the immunogen. So any agent capable of inducing an immune response is termed as immunogen. Okay, so there is a difference between an antigen and an immunogen. So antigen can be a immunogen, but it it is not hard and fast rule. Okay, so an antigen can also like initiate the immune response, but it is not every time true. Then all immunogens are antigens, okay, because they can bind to the antibody or the TCR, but they are not able, they initiate the immune response. But all antigens who are binding will not induce an immune response. Okay, now if we see any immunogen, so there are certain requirements that allow those immunogens to be actually active. Okay, so those requirements are: the immunogen must be foreign. Okay, then the size of the immunogen. For example, if it is less than 1,000 daltons, so those are basically antibiotics. So those are not immunogenic. Then, if there are 1,000 to 6,000 daltons, for example, certain hormones, so they either can be immunogenic or non-immunogenic. But the proteins that are greater than 6,000 daltons, like albumin or the toxins, those are sure shot immunogenic. Okay, then the chemical complexity. So if there are heteropolymers of something, for example, the proteins, they are heteropolymer of an amino acids. Heteropolysaccharides can be immunogenic, but homopolymers are generally non-immunogenic, or they induce a less immunogenic response. Okay, in terms of the biomolecules, we have proteins, the first, that are a good immunogen. Then we have carbohydrates, specifically the heteropolysaccharides, or those are complexed with the proteins. For example, glycoproteins or glycolipids, those are majorly the second number that can induce an immune response. Further, nucleic acid and the lipids are least immunogenic, although there are certain uh, lipids and nucleic acid that are recognized by the TLRs, but they are least immunogenic. Okay, then it actually sees that dosage and the route of administration. So the amount of the antigen that is going into the body, how much immune response it can generate. Okay, so obviously the high amount of antigen would induce a high immune response. Okay, then the route of administration. The subcutaneous route of administration has the highest propensity of inducing an immune response. For after that, the intravenous and the last is the oral route of administration. Okay, so these are some of the properties of a good immunogen that can initiate a good immune response. Okay, so moving on to another concept, that is heptane. So heptane are small molecules that when they are administered alone, they lack immunogenicity. Okay, but if an heptane is coupled to a carrier molecule, for example a protein, the immune response is induced. For example, here we can see these are the heptane molecules. When they are actually conjugated with the carrier protein, then it induces an immune response. Okay, then the heptane carrier conjugate, we have three antigenic determinants, either the antibodies to the heptane, antibodies to the carrier, or antibodies to the heptane carrier conjugate. So all the three can be generated when we inject a heptane conjugated with a carrier molecule. Okay, so why are heptanes necessary basically? So whenever we are in a vaccination program, okay, so giving heptanes along with the the major protein leads to induction of a good immune response and better memory, better immunogenic memory. Okay, so this is why heptanes are necessary, especially in the vaccination programs. Okay, so this is the diagrammatic representation of how a heptane can actually lead to the immune response. So as you can see here, these are the heptanes when they are given to rabbit, and then we test for the increase in the antibodies or the serum. So none of them increased. But when we conjugated with the carrier protein, then it actually leads to the increase in the antiserum or the increase in the antibody production. Okay, so this leads to the production of antibodies against the heptane, against the carrier protein, and both the heptane and the carrier protein both in conjugation. Okay, so if we have uh, but if we see when we this is the same thing that they are representing here. Okay, so whenever we do a protein testing for the heptane, that is DNP. Okay, so for example, so DNP will not be able to produce any antibody. Similarly, when we give a protein carrier, for example BSA here, it will lead to production of the anti-BSA antibodies. But when there is conjugate of both of them, so they will lead to the production of anti-DNP antibody major, then both of these are minor. Okay, now there are some heptanes that leads to their difference in the reactivity. Okay, for example, aniline, if we see, it is an aminobenzene. Okay, so the reactivity towards aminobenzene increases. Then orthoaminobenzoic acid, the it is the antiserum against this. Okay, same with the meta-aminobenzoic acid and the para-aminobenzoic acid. So the reactivity, whenever we give these type of heptanes, then the reactivity with those heptanes only will increase. Okay, so this is the case with the heptanes. So now if you guys have any doubt, you can ask, or else we'll start with the questions. Which of the following statements is or are correct related to the adaptive immunity? answer. Okay, let's see. So yes, let's see the options. Macrophages and dendritic cells link innate and adaptive immunity. Yes, this is correct. Okay. Innate immune cells do not express PRRs. No. Innate immune cells express PRRs as well as some non-immune cells also. Adaptive immunity is already present in the body during birth. No, innate immunity is present during birth, but adaptive immunity is acquired after birth. Innate immunity is highly specific. No, this is false. So, innate immunity is broad in nature. Adaptive immunity is specific in nature. The immune memory can be lifelong or short depending on the pathogen. Okay, so immune memory can be lifelong or short depending on the pathogen. Yes, this is correct. Okay, so because some of the viruses produce antibodies that can be lifelong, but some of the viruses actually produce antibodies that are short lived. Adaptive immunity is not passed from the parents to offspring, hence, it cannot be inherited. Yes, this is correct. Innate immunity can be passed. Innate immunity is generally inherited from parents and passed to offspring. Yes, this is correct. Innate immunity can lead to immediate and delayed hypersensitivity. No, this is the case with the adaptive immunity. Okay, so the correct answers are 1, 5, 6, and 7. Okay, so let's move on to the next question. Which of the following statements is or are correct? So, what can be the correct answer? 2, 3, 5, and 6. Okay, let's see the answer. So, T cells can bind to soluble antigens? No. B cells involve binary complexes of membrane, immunoglobulin, and antigen? Yes, this is correct. T cells require MHC molecule display processed antigens? Yes, this is also correct. Innate immunity has high potency? No, they have a low potency. Response time for adaptive immunity is slow? Yes, this is correct. Adaptive immune activates classical complement pathway? Yes, this is also correct. Adaptive immunity is found in both vertebrates and in invertebrates? No, it is a feature of the vertebrates only. Innate immunity has long term memory? No, it has no memory at all. Innate immunity can remember the specific pathogens? No, this is also false. So the correct answer is 2, 3, 5, and 7. So this would be 2. Okay, so let's move on to the next question. Which of the following best describes the role of heptane in immunology? So, what can be the correct answer? Okay, so let's see. A heptane can independently stimulate antibody production in a foreign host. No, a heptane will not be able to independently induce a immune response. Heptane can only induce antibody production when conjugated with a carrier protein. Yes, this is correct. A heptane lacks both foreignness and an immunogenicity. No, they cannot be considered a foreign material. They can be host specific also. A heptane directly induces immune responses in animal without modification. No, this is also false. A heptane is structurally identical to an immunogen but has lower molecular weight. No, this can also be false, because if we are talking about an heptane, that is not considered as an immunogen at all. Okay, because if there would be some immune response that it can generate without the carrier protein, then it will be considered as an immunogen. Okay, but when we are dealing it with the carrier protein, that means it will not be able to induce an immune response alone. Okay, so the correct answer is option number 2. Okay, so let's move on to the next question. Which cells are known as professional antigen presenting cells? So what can be the correct answer? Yes, the correct answer is option number 4, the macrophages and the dendritic cells. So these are the professional antigen presenting cell that phagocyte the unwanted cells, and then they present it to the MHC, to the T cell receptor. Okay, so this was the last question for today. So if you guys have any doubt regarding anything, you can ask. Okay, then we'll close the session for today. Thank you guys for joining in. We'll meet next week.